enzymes in biological reactions?
By Sebastián Miranda and Asa Quevedo.
Water enzymes. Enzymes are biological catalysts made up of long chains of amino acids which help speed up the chemical reactions in our bodies without being consumed. Every chemical reaction depends on our enzymes, and too little or too much enzymes can cause problems to our bodies.
How do enzymes work? One of the most important roles of enzymes is to aid in digestion. Digestion is the process of turning food that we eat into energy. That's the main role of enzymes in our body. For our bodies to do reactions, we need something called activation energy. Enzymes work by lowering this energy, making the reaction happen faster and efficiently. If there would not be any enzymes, reactions would be too slow to actually sustain life. Their unique shape determines their function, particularly in their active site, which is where the reaction occurs. The active site is specific to a particular molecule called a substrate. When the substrate binds to the enzymes help it convert into a new molecule called the product. In this example, we see a substrate that are two molecules bonded together, binding with the enzyme. The enzyme breaks the bond, having two products. Some examples are the enzymes in our saliva, pancreas, intestine, and stomach. They break down fats, proteins, and carbohydrates. Enzymes use these nutrients for growth and cell repair. Example 2, the enzymes sucrase breaks down sugars called sucrose. Lactase break down lactose, a kind of sugar found in milk products. Another great example is a catalyst, an enzyme that helps break down hydrogen peroxide, H2O2, into water and oxygen.
Competitive inhibitors. So competitive inhibitors are molecules that have very similar shape or structure to a substrate. They bind into the enzyme's active site, blocking the substrate from attaching to the enzyme, making the reaction slower. In other words, they lower the reaction rate by reducing the number of enzyme substrates attaching to the enzyme. However, this effect can be reversed by increasing the concentration of a substrate. Non-competitive inhibitors. So non-competitive inhibitors are molecules that bind into an enzyme at a location that isn't the active site, known as the allosteric site. This changes the shape or function of the enzyme, which still prevents the substrate from effectively attaching to the enzyme.
How does pH levels and temperature affect the enzyme? The pH. Enzymes are sensitive to acidity and alkalinity, and they don't work properly if in an environment that's too acid or too basic. For example, an enzyme in the stomach called pepsin breaks down protein. If your stomach doesn't have enough acid, pepsin can't function optimally. In temperature, enzymes work best when your body temperature is normal, about 98 degrees Fahrenheit or 37 degrees Celsius. As temperature increases, the enzyme's reaction increases. But if the temperature gets too high, the enzymes stop working. That's why a high fever can disrupt the bodily functions. What are the different types of enzymes? There's the carbohydrates. Carbohydrates are enzymes that break down carbohydrates into simple sugars for digestion and energy. For example, the amylase, which breaks down starch into maltase, which converts maltose into glucose. Lactase, which digests lactose, and sucrase, which break down sucrose into glucose and fructose. Those enzymes are essential for absorbing nutrients and fueling your body. Another type of enzyme is the prostate. The prostates are enzymes that break down protein into peptides and amino acids, which the body uses for growth, repair, and energy. This is the enzyme that most teenagers have growing up. They are found in the stomach, pepsin, pancreas, the trypsin and chymotrypsin, and the small intestine, the peptidase. Prostates are essential for digestion, muscle maintenance, and immune function. Another type of enzyme is the lipase. Lipase is an enzyme that breaks down fats, lipids, into glycerol, and fatty acids, making them easier for the body to absorb and use for energy. It is mainly produced in the pancreas and works in the small intestine. Lipase are essential for digesting fatty foods like oils, butter, and dairy, helping the body absorb fat solubles like vitamin A, D, E, and K.
Thank you for your attention.